Okay, so once your body has detected that something out of the ordinary is happening, either that there is an invader or that damage is occurring, what does your body do? And so now we're going to start looking at responses, and of course for right now we're looking at the responses associated with your innate immunity. So we're going to look at how does your body respond to danger. And some of the different responses we're going to look at your complement system, we'll look at interferon production, we'll look at phagocytosis, and inflammation, and fever. So we're going to look at each of these responses one at a time. And I'm going to start with complement. So the complement system is a system of about 30 different proteins that are often called complement 1, complement 2, so the letter C for complement and a number, or they're also called factor A, factor B, but they are proteins that are made by the liver and they circulate in your blood, but when they circulate in your body they're in an inactive form, and the only time that they become active is if they either directly or indirectly encounter microbes, and then that will activate them and trigger off the complement cascade and complement responses. Their job is to enhance the immune response, and that is how they got their name. When they were first discovered, they were discovered to be working sort of with antibodies, and so it was said that their action complemented the action of antibodies. So that's how they got called complement proteins. And what we're going to see is that there's different ways to activate complement. So these three boxes up at the top of this picture represent 3 ways to activate complement, but however you do it, ultimately where you end up is activating this key complement protein, C3. So C3 is the key complement protein because it is at sort of the intersection of all the activation pathways, and it's the first step in all the responses, which are what you see down at the bottom. And so C3 in its inactive form consists of C3a and C3b joined together. But if you split C3, if one of these 3 activations causes C3 to split, then you get these proteins C3a, C3b. They are active. They can in turn split other complement proteins, and you get this cascade of effects. So we're going to look at each of these different types of activation, but you don't need to know all the steps. You don't need to know which complement proteins are involved. You just need to know what is the first thing, what is the key trigger for this particular type of activation. So for the classical pathway, this was the first one that was discovered. It is antibodies, and in particular it's antibodies who are attached to the surfaces of a microbe. So antibodies are proteins. They have little recognition sites where they can detect antigen, and they will bind to the surfaces of microbes, but when they bind they change a little bit, and what happens is the back end of the antibody becomes changed so that it can interact with other things, and one of those things it can interact with are complement proteins. Okay, sorry, I paused there and I kind of forgot where I was, but the complement activation pathway that's called the classical pathway involves, it was the first one discovered, it involves bound antibodies interacting with complement protein number 1, and I know I already said that, right? And that leads to the splitting of this protein, and again, you do not need to know the details, but look at how this leads to ultimately the splitting of C3. Okay, so remember that the classical pathway, it's bound antibody that interacts with complement protein C1 and leads to the activation of complement. The second pathway that was discovered was called the alternative pathway. They had the classical pathway, so now here was an alternate. And this is triggered by cell wall polysaccharides. So you have microbes that can have either the O polysaccharide if they're gram-negative, or maybe they have polysaccharides in their glycocalyx, and these polysaccharides interact with some complement factors that then lead to the splitting of C3. So polysaccharides kick off the alternative pathway. And then a more recently discovered pathway, the lectin pathway, there are proteins from the liver called lectins that bind to certain sugars on microbes, these mannans. And when the lectins bind to the mannans, the lectin's shape is changed so that they can interact with complement proteins and ultimately lead to the splitting of C3, right? So all three pathways, classical, alternative, lectin, they lead to the splitting of C3. What you need to remember is what is the key thing that triggers each of those pathways. Once C3 is split, we have these activated units, C3a and C3b, and then they do different things that lead to the outcomes of complement. And the outcomes of complement, of course, are targeted towards getting microbes out of your body. Now, from here on out, I do want you to know the specific complement proteins that are associated with each of these responses. And the first one we'll look at is opsonization. And so this is mediated by complement protein C3b. And so when C3 is split into C3a and C3b, one of the things that C3b can do is stick to the surface of microbes. And so all those little tiny red dots are supposed to be C3b stuck to those microbes. And that's handy for you because phagocytes have receptors for C3b. So if something has C3b stuck on it, it makes it easier for the phagocytes to bind to it and grab it. And this is especially important when you're looking at things like microbes that have a glycocalyx, where maybe the phagocyte can't bind to the polysaccharides in the glycocalyx, and so you have this slippery microbe that's getting away. Well, if complement is activated and C3b is stuck to the microbe, that gives your phagocyte a handle so that it can grab on and bind that slippery microbe and digest it. And this is called opsonization. Anytime you have something that's stuck to the surface of a microbe that helps a phagocyte bind, we call it opsonization. Complement is also involved in enhancing the inflammatory response. So C3a and C5a bind to mast cells and basophils, and when they do, they cause degranulation. They cause those little vesicles of histamine to go up to the plasma membrane, fuse with the plasma membrane, and release histamine outside of these cells. And then the histamine acts to trigger inflammation. So they release histamine, and that increases the permeability of blood vessels and triggers inflammation. C5a is also a chemoattractant for phagocytes. So having C5a in the neighborhood will recruit phagocytes to the area. And then C5 also participates in a response called cytolysis. So C5 interacts with C6, C7, C8. There's kind of a chain reaction that happens. And you end up with these complement proteins inserting themselves into the membranes of bacterial cells. So they form this ring-shaped structure, and C9 punches its way all the way through a membrane. So you get these little soda straws again. So here's a picture of a cell with lots of these protein channels embedded, and this whole little complex here is called the membrane attack complex, or MAC. And so when MAC inserts itself into membranes, that lets cytoplasm leak out and causes cells to break down. All right, and so I'm going to post the animation on your Canvas site because I think it's better to watch it with the narration. But I do have this little movie that shows the MAC complex in action. So in this movie, what they did was they took red blood cells, and the reason they used red blood cells is because they're big and they're red and you can see them, and they mixed them with antibodies to those red blood cells and complement proteins. And so if you pretend for a minute that these red blood cells are actually bacteria, you'll see how the MAC complex helps you, right? So if these were bacteria and MAC is setting up in their membranes, right, they're all exploding is the point, right? So that it kills the cells by making them explode. We'd see only the ghosts of cell walls left behind if it was bacteria. Now, this would not happen in your body, assuming you didn't get an improper blood transfusion, because you should not be making antibodies to your own red blood cells. So there should never be a situation where you have antibodies triggering complement reactions to your own blood cells. Okay? All right, so I'm just going to go back and recap, go back to this big picture, right? So we looked at 3 ways to activate complement. We saw the classical pathway, which is activated by bound antibodies, the alternative pathway activated by cell wall polysaccharides, and the lectin pathway activated by lectin proteins from the liver when they bind to sugars found on microbes. All 3 of those pathways, you have an interaction with a complement protein, activating that complement protein, which activates another complement protein, some kind of little relay, and ultimately you activate C3 and split it into C3a and C3b. We saw that C3b helps with opsonization, C3a enhances inflammation, C3b also can split C5, leading to C5a, also enhances inflammation, and starts that membrane attack complex, which punches holes in the membranes of cells. And so the complement defensive system is responding to the presence of microbes and leading to reactions that help defeat microbes when they're inside your body. Okay, that's it for complement.